everybody. My name is Wolf Geppert. I'm the chair of the European Astrobiology Institute, and I would like to welcome you very heartily to this seminar, the first one in 2021, which is given by Heather Graham. Heather is an organic chemistry, and she is also involved in the ENFOLD research network of NASA. She has a PhD in geosciences and geochemistry from the Penn State University from 2014. After that, she got the NASA postdoctoral program fellowship and is now on a Canadian Institute for Advanced Research fellowship. Her talk and her main research field now is about agnostic biosignatures, and that's what we are hearing today. So welcome, Heather, welcome you all, and the floor is yours. Thank you very much for coming.
Thank you, everyone, for letting me kick off your 2021 seminar series. This is really exciting, and I really want to thank Wolf for inviting me. Part of what I'm going to be talking about today is my research and the research groups around the world that I'm involved in, as well as a little bit of the work of the Network for Life Detection and ways that we can collaborate between our two research groups, the EAI and ENFOLD. I'm assuming everyone can hear me okay. And if there's any problems with the slides, just let me know. So yeah, so we're going to start out with a little bit of what ENFOLD is, what it means when I say agnostic biosignatures, an overview of ongoing projects in the Laboratory for Agnostic Biosignatures, which is an international consortium of 40+ researchers in 14 countries around the world. I'm the deputy principal investigator for that effort, and I'll talk about the work we're doing. And then I'd like to end with some discussion of methodological hurdles and knowledge gaps that we have when we talk about agnostic biosignatures and biosignatures in general. And this is an area where there might be some really fruitful ways that ENFOLD and the EAI can collaborate in the future. To start off, ENFOLD is the Network for Life Detection. It is one of the newly minted research coordination networks that was founded by NASA over the past couple years. There are other elements of these research networks, including one that's focused on exoplanets, one that's focused on prebiotic chemistry, and another one focused on ocean worlds. Since I know that research coordination networks mean a different thing for pretty much every organization, what we mean is it's a virtual structure to support collaboration between investigators. They help us plan research, training, and educational activities, and give us some support for that sort of activity. So Wolf and I have been talking about possibly in 2022 with Beacon, having an in-person coordinated effort between ENFOLD and the European Astrobiology Institute, which I'm really hoping can come to pass. ENFOLD is made up primarily of 3 core groups, including the Center for Life Detection Science at NASA Ames, and Oceans Across Space and Time, OAST, at Georgia Tech, and of course the group that I'm deputy for, which is the Georgetown University's Laboratory for Agnostic Biosignatures. We do have many other affiliate members from other funded NASA efforts as well, and include members like Wolf and possibly others in this group who don't have NASA funding but do participate in NASA-related research or collaborate with NASA researchers. So our objectives, like I said, are to think about biosignature detection strategies, develop some methods of collaboration. This is a long, exhaustive list of some of our goals, but the two that really relate to my work are the organization of a reference sample set, which we're hoping can be an international community resource at some point. I'll talk about that towards the end of the talk. And then, of course, ways of engaging the community with the ladder of life detection, which is something I'll discuss, and incorporating the ideas of agnostic biosignatures. So that gets us to this idea of agnostic biosignatures. What does that mean? And when we talk about this, we need to consider, how do we contend with the truly alien? How do we search for indicators of life without making assumptions about what we think will be in the molecular framework of any particular biochemistry when it is unfamiliar to us and possibly does not share a common heritage with Earth? So agnostic biosignatures, just so that everybody remembers what we mean when we say biosignature, that's just a sign, a physical sign that there was life. It could be something as simple as a footprint or as complicated as a hopanoid in a many million-year-old rock. But when we're talking about an agnostic biosignature, we're meaning those that are not tied to a particular metabolism or informational biopolymer, or really any chemistry that is a characteristic of life as we know it. And I always want to stress to people that this isn't just one single thing that you can find. It can be a system-wide alteration of a planetary environment or a preservation environment in the geologic record. So it can be one thing, or it can be a community of indicators that tell us that there's a very different biochemistry at work in some substrate that we are interrogating. This work was inspired, our group was inspired to form based on some recommendations made by the National Academies in the 2016 Astrobiology strategy document. I know there were many European collaborators in this work, so I wanted to thank any of you in this group who might have participated. And there was a very strong recommendation that was repeated many times that said that the group felt that it was really essential for the search of life beyond Earth to require frameworks for non-terran life, and that we should start to really think about novel or agnostic biosignatures, both here and on other planets. So I'm sure there are many of you who are engaged in more traditional biosignature activity, either here on Earth or on other planets, and this might be a curious idea to all of a sudden start trying to reframe all of our work. And I would say that for someone with a background in Mars research, life as we know it and thinking about similar biochemistries is a great starting point for when you're talking about Mars. It's nearby, it has very similar geologic history. It once looked very much like Earth, had water and a magnetic field. So there's a strong possibility that any life on Earth could have had a common ancestor with Mars. But that whole idea is a little bit harder to relate to other planets in our solar system. We have lots of other good reasons for using typical life detection methods based on our own geologic record, and that's that they are very robust. We have spent a lot of time as a community, especially in geology, relating all of these very particular chemical and physical signatures to life that we can either look at directly that's extant, or find relatives that are still extant here on Earth. So this sorts of things is the patterns and particular molecules and isotope signatures that we associate with life. Patterns within those molecules. I don't know if you can see my cursor, but this would be a pattern that we are talking about when we're thinking about a population or a community of molecules. Biogenic structures and textures and biogenic minerals, those are all familiar to us from current life. And so we know how to interpret that data. We know where it's starting from. It's a signal that's rich in information. And most importantly, we really understand taphonomic processes. We understand what time and pressure does to these chemical signals. So these are all great reasons to not abandon our traditional biomarker studies, but really start to think about the general features of them and how they actually relate to other solar system bodies. So yes, as we go out into the outer solar system where there are lots of habitable worlds, the many moons around the ocean worlds, we have to start thinking a little bit differently, because the likelihood of common heritage between life here on Earth and life far out in the solar system is greatly decreased. However, there are many habitable environments that are similar to Earth environments that we can interrogate and develop biosignature systems for. So when we start to think about agnostic biosignatures, I always like to tell people, we're just looking for the more general characteristics of life. A lot of the things that we talk about in very traditional biomarkers, like hopanoids or things like that, are functional molecules or complex molecules, things that we know how they relate to metabolism, and we have very specific information about their expression and their origin. But when we want to think about agnostic biosignatures, we need to think about the really basic features of life. I often like to tell people that agnostic biosignatures isn't necessarily about biology, but it's more physics detection. We're looking for signals in very basic features that don't arise without the influence of extra energy in the form of life. I don't know if you guys are familiar with this framework that we are encouraged to link our work to here in the US, and it's called The Ladder of Life, and it's based on a publication by Mark Naveau in 2018. And I'm sorry I don't have that reference here on that slide. I meant to put that there. So yes, refocusing on the less accessible but more possibly fundamental features of life and challenging those assumptions about what is universal. I think this is also a way that we can start to really challenge our ideas of the limits of analogy. As someone who used to work on the Earth's geologic record, I always tell people that it was easy for me to make the pivot into agnostic biosignatures, because when we look deep in the Earth's record, we find life that's impossibly different than what we see now. And so to understand life in deep time on Earth, we also have to go through this process of divorcing ourselves from our expectations and really thinking about the limits of analogy for current extant life on Earth. And this leads us to these guiding questions we have in our group, the Laboratory for Agnostic Biosignatures. Are we interrogating the full possibility space for life? This is a lot of the reason why we have a strong theoretical component in our group. Are we thinking of the spectrum of possibilities for life as we don't know it? And something that we're always talking about is, how would we have designed the Viking experiments differently if we did them today? And this is just a way of recognizing that our expectations for life and habitability and that possibility space has changed dramatically in the 40, 50 years since we designed those experiments to be done on Mars. We have a very different expectation for what might actually exist there and what metabolisms would be possible. So we're, while we're thinking about applications on Mars, we're really focusing our future work on Titan with the idea of supporting the Dragonfly mission that's going to be launching in, I think 7 years, which seems very, very soon. So yes, the Laboratory for Agnostic Biosignatures is primarily 4 research groups focused on complexity of molecules, complexity of surfaces, chemical fractionation, which is just patterns of elemental distribution, and disequilibrium and energy transfer. Within these 4 groups, we also have a theoretical group, which supports all of our work as a cross-cutting element and provides computational resources to everyone. We also have a benchmarking and curation group, which provides common samples to all 4 of the theme groups, so that whenever we are examining a potential analog for a planetary substrate, we're doing it in coordination with all of the other methods that are under investigation. So we're primarily thinking about tools and techniques for flight instrumentation. We're a big group. I wanted to point out that we have a very large contingent at the University of Glasgow involved in primarily our molecular complexity component. And we also have a theoretical partner at Umeå University in Sweden. And he supports molecular complexity, molecular complementarity, and chemical fractionation, all of those theme groups, as well as engaging in some of the specific tasks for that theoretical and computational team as well. So I'm just going to go through each of those 4 groups pretty quickly and give you guys an example of some of the research that we're involved in. The first group that I was going to talk about is the chemical complexity group, and that is just this idea of thinking about the intrinsic information embedded in a chemical structure that tells us the likelihood that it would form without the extra energy that's provided by biology. So this is the idea that there's a threshold above which a molecule is unlikely to form in an abiotic environment. We have a very strong component in our group of people involved in meteoritics. So we're really trying to embed ourselves in the understanding of abiotic organic chemistry, both in the deep earth as well as out in space, since that is the inheritance that we're seeing for most of our nascent biological systems on other planets. So I'm going to go through this idea of molecular assembly complexity that guides our work. I wanted to let you guys know I'm going to talk about it a little, but I don't feel comfortable showing the graphs quite yet. But we do have a new publication in review. It's on Chem Archive, and I'll give you that information as well. So the molecular assembly complexity index is this idea that there's a non-random abundance in complex molecules that distinguishes it from abiotic organic chemistry. These are all organics. We're not talking about minerals for this type of index yet. So based on this, we're understanding that this is really linked to the fact that living systems evolve and allow generation of complex molecules. This goes back to a lot of the work of the theory group that we have at University of Arizona as well. So just to walk you through that, it's this idea that every time you add in a new component of a molecule, you're not just thinking about molecular mass, but you're thinking about, is that just an iterative process? This is not necessarily adding new information if we're just stringing together carbons, because each of those are basically monomers of the last. Whereas if we start adding in branching, cyclicity, heteroatoms, things like that, we're drastically changing not only the polymeric features of that molecule, but the likelihood that it would be formed abiotically. So you can think to yourself about the kind of things we see in space, like giant PAHs, versus the very particular molecules that we associate with life, like a hopanoid. So the way that we're starting to interrogate wider chemical space is by using algorithms, because we can't possibly look at all of these molecules and their structures independently on our own. And we're using these ideas from graph theory to guide this idea, again, of how do you build up. And when we find an independent building step, we number that. And so this is really a way in molecular assembly complexity index of counting those steps that are each unique. There's lots of different ways, for example, that you could have built biphenyl, but it really comes down to this 6-step construction process. Then we start to look at how that relates to other common ideas that we have about molecular complexity. And when we start to relate molecular weight to this step process that we've borrowed from information theory, what we find is that above a certain threshold, all of the molecules that we're seeing above a certain number of steps are only derived from biology. Maybe they aren't natural products, but they might be synthetic drugs or things like that, which we would think of as a technosignature in the chemical world, something that's highly unlikely to arise on its own, but structividin or something like that. These sorts of commercial products, but are related to our biology since we made them. So one of the things that you'll find if you look up this paper that we have on Chem Archive that we're hoping to come out soon is that we have been really trying to relate this molecular assembly index, which relates to the likelihood that something was made by biology directly or indirectly, to the number of fragments in tandem mass spectrometry. And we find an 89% correlation between molecular assembly number and the number of fragments. And you might be thinking, well, that's a really silly sort of correlation to make, but the reason that we're really focused on this connection to fragmentation is that this is a really important tool that we have for interrogating chemical systems on other planets. This is really what this instrument, the MOMA instrument, the Mars Organic Molecule Analyzer, mass spec, can do for us. This is an instrument that's being built just down the road from me at Goddard, where I'm normally working instead of my house. And this is an instrument that will be on the ESA rover, Franklin, that's going to be leaving in next year. And it's also basically the same instrument that Dragonfly will be taking to Titan. So this is, again, that idea that we're going to be looking at things on Mars as well as an ocean world. So we're really trying to expand our understanding of agnostic biosignatures using this fragmentation framework. So fragmentation mapping is basically complexity applied. I have here on this side 2 molecules with wildly different complexity metrics, 16 and 7. So that tells you something about the likelihood that they would be made abiotically. Biology, of course, generates higher complexity molecules. And these molecules have higher relative abundance than lower complexity molecules in biological systems. And those higher complexity molecules will produce more ions during this fragmentation, like we can do with these instruments we're sending to other planets. So this is just an example of some of the data that we see coming from the MOMA instrument, this flight-capable ion trap. And right now we're doing a lot of work with the test bed that we are building as part of Laboratory for Agnostic Biosignatures to really robustly test how the MOMA-like instrument that we're building will respond to a variety of these kinds of complexity biosignatures on Mars as well as eventually on Titan. So here's another example of us looking at abundance patterns versus the kind of fragmentation mapping that you'll see. And of course, the ones that are biologically derived are in greater abundance and higher complexity than those that we would expect just in, meteoritic output or abiotic organic chemistry. Oh, it looks like I accidentally repeated some of that information here, but I just wanted to show again that this is that idea of the abiotic versus biotic when you're thinking about complexity distributions as well as the distributions of the abundance of those molecules. So this gives us some ways of also thinking about populations of molecules in a computational sense. This is an activity that's very familiar to people who might work in the geological record, where we're using statistics, or even ecology, where we're using statistics to think about populations of mixtures of molecules. So if you had a mixed abiotic-biotic system, so we're using chemometrics to try and separate in statistical space these molecules from each other, and we'll use those computational tools as well with the kind of data that we're getting from this flight instrument mass spectrometer. The second theme that I wanted to talk about is also based on complexity, but this isn't molecular complexity, this is surface complexity. And this also has a statistical background, kind of like the chemometrics in molecular complexity, as it is going to be applied by flight instruments as well. This is the idea of using 2 high heritage techniques, sequencing and SELEX, to map the surfaces of substrates that we find in liquids on other planets, or in potentially think of deeper brines or something like that on Mars. And the idea here is that if you take a whole bunch of randomly generated aptamers, so those are oligonucleotides, strings of DNA, can be as short as 15 or as big as 80, and you allow them to interact with a surface, because they have many different ways of folding and creating their own binding structures like antibodies, they will bind with these complex mixtures in different ways. So what I'm showing here is if we took a sample, say this is an Enceladus plume, and it has cells as well as minerals, and we applied a population of randomly generated aptamers to it. In the cell, you would have many binding, and on the mineral, you would have few binding. And we know that oligonucleotides or aptamers like this do generate binding specificity and affinity, just like antibodies, and bind to a variety of materials. We can think of them for understanding molecules themselves or surfaces, even inorganic surfaces or metals. And we know that there is binding that occurs between aptamers and all of these things. Like I said, this is borrowing on high heritage from other techniques. So if you're familiar with the idea of CellX, it's a way of doing some in vitro selection of an oligonucleotide that is highly specific to a surface. So in CellX, you would take a random pool of aptamers, you'd let them incubate with your surface. In this example, it's a cell, but if we're talking about an unknown, it could also be just a particle. We remove the unbound sequences, and we collect those that bound. We test it with something to see if there's unspecific binding, and then we amplify those and look at the sequencing. What that sequencing tells you is what the binding properties of that unknown random aptamer were. For that, when we think about flight applications, we can think of nanopore sequencing. This is something that has been tested on the ISS. We think that it works in space just fine. And it's a way of doing native DNA sequencing. You don't have to tear it apart to sequence. And it would be able to tell you what the identity of that randomly generated aptamer was, so that when you apply it to a surface, be it biological or abiological, you get the sense of the chemistry of that aptamer and how it bound. This is a way that we can interrogate low biomass targets, since first of all, it's not destructive, right? You're exposing your sample, say it's your Enceladus plume material, to your reagents and your aptamers. But then what you're doing is you're removing all of those aptamers that bound to it and then amplifying them up. So this is another example of us using a high heritage technique from biotechnology. So we're able to take life that probably has very, very low biomass, like we expect with chemolithotrophic systems in the ocean worlds, and have demonstrated in similar systems in the subsurface, and amplified them up to look at what the signal from the binding nucleic acids was. So this is another example of us using statistical analysis or chemometrics, where we're looking here at the binding of aptamers on certain objects. These were filtrates, and seeing that they do clump together in statistical space based on whatever that sample was. We're also improving on some of this aptamer binding by trying to optimize a proximity ligation assay. And so this is a way that we can be a little bit less specific with the kind of aptamers that we're trying to make bind to our surface, and also gives us something that can be amplified directly, since it has these primer sections on it. So this gives us a very analyte dependent interaction that we're going to amplify. And there's this is a lot of the current work that we're doing, is really trying to optimize this PLA technique. And we've been doing mapping of the chemical surfaces of a number of different things, including magnetic beads that have been coated with antibiotics that are typically used in biotechnology labs, as well as filter surfaces, celluloses of different kinds, glasses of different kinds. And so really trying to understand the differences between those organically coated metal beads and something that we know is abiotic, like a glass or a metal disc, and then that kind of extreme complexity that we see in celluloses. There's different ways that we'd like to possibly modify this in the future that wouldn't necessarily rely on sequencing. And this is the idea that if we did just have a population of these aptamers that we applied to samples, say in a ocean world's liquid sample, we could just use a fluorescent probe. And the value of this is we wouldn't have that energy and computational processing needed for sequencing. And it's something that could be image-based, so it would be very low data, amounts of data that we would have to collect through, transmission from the outer solar system, which is a problem. The third group that I wanted to talk about is our chemical fractionation group. And this is a similar way of thinking of mapping surfaces, but in this case, we're looking at elemental and isotopic accumulation on surfaces as a way to basically spot biology. You could think of this. These are cells that were taken from filtrates from waters in the deep subsurface as basically being a way to look for a cell by looking at the typical either elements or combinations of elements that we find with life. And what you'll see is that this is a way that you can immediately start understanding particles that are biotic versus abiotic, since we do see accumulation above background of isotopes and elements that we wouldn't see if it was only physics that were generating these particles. This is another way of relating work that we can think of on very large scales, where we're looking at large biogeochemical patterns, such as the Redfield ratio. And that's the typical carbon to nitrogen to phosphorus ratio that we see here on Earth, that's nearly constant across the Earth, with little bitty tweaks that we think are related to metabolism. That is an elemental abundance being used in a very coarse way, admittedly, as a biosignature. But it's more of a system-wide approach, as I alluded to earlier in my introduction. Instead of an individual way of thinking about a singular biomolecule, this is a way of thinking about it on a population basis. This idea of the Redfield ratio, though, is still observed at the microscale. Life is concentrating scarce elements that we typically wouldn't think of finding in their cells, and those become discrete, geochemically distinct, quasi-stable entities. And that is, I guess, what a geochemist might think of as a cell, is that concentration of elements. So here we're showing at least 10 elements that are enriched substantially in these cells versus the abiotic precipitation of calcite. So if we were interrogating these two particles and not making an assumption about which is a cell and which is just calcite, we would be able to see these patterns just in their elements, even without thinking about the surface complexity from the last theme or the molecular complexity of the first theme. We have some new work coming out that we've done with our theoretical group to really dig into what is generating these signals that we see in observed ratios of important elements for biology. And something that I'd like to point out here is that if you think of cell volume and the N to P ratio, when we're looking at the oceans and we're looking at the Redfield ratio, we're really seeing an overexpression of these most common middle-sized cells in the ocean. And if you look out at the other ends of the size spectrum, you see a very different N to P ratio. So if you were in a different ocean where there were different sizes of cells that were being favored, you might have a very different ratio to think about. Oh, and I should mention this, this paper is probably going to be coming out later this month if anyone's interested in this connection. We're doing a lot of this work in subsurface mines in Canada and South Africa, where there's chemolithoautotrophic systems that have been out of communication with the surface of the Earth for sometimes billions of years, which represent a pretty good analog for thinking about an ocean world, which is similarly going to be very different heritage than Earth, and also very energy limited and probably very, very low biomass. So we have these biosamplers that we're using for in situ colonization in these boreholes. And here's just some images from something that's a little bit more accessible, the Eel River site. And here you're seeing an example of where we can map out where the cells are based on elemental and isotopic abundances without thinking about any other considerations necessarily of biology. Our final theme that we think about is energy transfer, really digging into this idea that life is nothing but an electron looking for a place to rest. That if you see energy disequilibrium in a system, there will be a consistent way of identifying if it is abiotic versus biotic. This is some work that's being done by our group at Harvard, and they have been deploying these electrochemical sensors in the deep subsurface of marine environments to try and detect redox reactions that do indicate active metabolism and are probably most likely related to EET, extracellular electron transport. And here you can see in an inoculated bioreactor that you get very different energy transfer patterns when you add in organisms that perform this EET versus bioreactors where that is not happening. So this is the basis of those biosensors that they're using in the ocean, is a conductive electrode that looks for those electrical properties that we associate with biology. So life using solid phase minerals as electron acceptors. Here's an image of that sort of apparatus in the seafloor, looking for biomolecules that are redox active. And this is another area of heritage where we know that organic molecules in liquid media and how they do respond as opposed to abiotic redox active species. So molecules, yes, tend to have their own electrochemical signature, and we can relate that back to their likelihood that they are biological. So feeding into the other themes of the group. Here are some examples of our test sites in deep mines in the Kidd Creek system in Canada, where there's a very fresh borehole that's been drilled, and we are hopefully next month going to be putting in place one of these electrochemical sensors, as well as one of our filtering systems, so we can do some broad theme work with these materials and test across all of our theme groups how these filtrates and how these waters relate to our expectations for agnostic biosignatures. I wanted to also remind everyone that we have our computation and theory group, that everything we do is incorporating computational work to guide us through theory about life that we don't observe. We're trying to be very true to that idea of observed versus theoretical life. So that was a good example of that was in our N to P ratio work. Another big job of the computation and theory group is building this Bayesian network. Like I said, every single group looks at a common set of samples, and we're building from that expectations, not of yes life, no life, but a probability that something is physics related versus life related. And part of this group as well is the curation and benchmarking group that goes out and collects novel samples, both extraterrestrial, extant, geologic samples, inorganic, a wide variety of things, and we're trying to build up this database so that we have a really good sense of pairings of instruments that work well together from the 4 theme groups that we're studying. The way that we're expanding this out is through the N-fold network, where, like I said, we're interested in pairings of instruments because we don't think any one piece of evidence is ever going to convince anyone that we found life off of Earth. And our 4 ideas are certainly not exhaustive. There are many other people using interesting biosignature systems and interesting agnostic systems even, that we can start to compare how our methods work together in different systems with likely planetary analogs by including them in this sampling and analysis scheme. So this brings me to some of the related topics that are salient to NFold, and what I'm inviting all of you to think about here today with me, and I hope we have a chance for discussion, as well as in your own network and your own individual research. So this is potential paths towards agnostifying your own life detection method. This isn't a good or bad thing, like I said. This is just a way of really trying to understand the possibility space for any method that you might be working with. Something that we're really trying to get people thinking about is how agnostic approaches require a different kind of contamination knowledge regimen when we're thinking about flight instruments. And finally, a challenge for a better understanding of abiotic organic chemistry. So going back to that idea of analogy, making sure we're looking for life and not just appropriated patterns from Earth. I always like to throw an Andy Warhol painting in here, because so much of his art was about thinking about appropriated patterns. And a lot of times when I'm talking to people about biosignature systems, I'm aware that in many cases we're just taking patterns we're familiar with from life on Earth and trying to kind of basically wiggle match on other worlds. And being really aware of when you're doing that you're narrowing your search pattern and making assumptions about the heritage of that living system, should it exist. The other thing is using these search patterns to consider cryptic communities. A lot of our work that's in the deep subsurface is in places that, like I said, have not been in communication with the surface of the Earth for 2 billion years. If there are organisms there, they have been on a very different evolutionary path than us, and that's really making it hard for us to find life in these systems. We have all these geochemical indications that there's metabolic processes, but it's very hard to find those cells, possibly because the primers that we use just aren't, related to the kind of heritable material that these organisms have, because they've been on a very different evolutionary path. So this is useful for cryptic communities here on Earth, as well as on other planets. And being aware that the current contamination controls that we have are heavily biased to contend with terran life. Here's kind of an idea of thinking about where universal biosignatures or agnostic biosignatures relate to terran biosignatures, which is the typical biosignature work that most of us have been engaged in for a long time, and questioning those limits while targeting universal things. This does trade specificity for inclusivity, but that also means that you do have to build a network of observations, which is why we have the paired sampling system and analysis system that we have. Yes, again, I think I already said this, but be aware that the identity and behavior of potential contaminants might be your biosignature. So this isn't a bad message. This is just something saying that we just need to know more about what we send with our instruments, so that we know when the signal that we're looking at is just us. And then finally, this idea of better coordination with abiotic organic chemistry. Especially, we think about this a lot with our tandem mass spec method, since many of the polymers that we see in meteoritic materials give rise to regular fragments, and this might initially make us think that there's some sort of metabolic process that's built on polymers, but really this is just a fragmentation artifact. So with that, I wanted to just once again say welcome to NFold for those of you that are involved. I'm happy to talk about the network, happy to talk about ways that we can collaborate. I with the steering committee, as well as my duties as the Deputy Principal Investigator for the Laboratory for Agnostic Biosignatures.
Okay, many thanks to to header. And now we have the possibility to ask some questions. So if you want to ask some questions, you have got two possibilities. Either you switch on your video screen and wave a little bit or something like that, or just switch on your video screen to ask a question, or you put it into the chat and I read it out of you. I actually have a question from Floris van der Tak now.
Hi, Heather. Greetings from Groningen, the Netherlands. That was a great talk. Thank you very much.
Oh, thank you.
The question I had was about the second of your themes. So this is the surface complexity. I didn't really get what kind of surfaces you were talking about. Just the surface of a rock or a mineral?
Oh, yeah. So we're looking at both. We're trying to understand the inherent complexity in both. Our idea is that something that has very regular chemical features, like a mineral, will have a limited number of binding sites when compared with the kind of ornate chemistry you see on cells. So cells have all sorts of transfer proteins and different molecules embedded in the membrane to change properties of the membrane. And so all of those things represent new kinds of binding chemistry that are possible. So that is what we're really testing with our selection of samples that we're using with that proposed instrument right now, is just understanding how much more diversity of binding do you get with a cell versus a mineral? So the answer is both. We're looking at both right now.
Yeah, and so that will be on a submicron scale, I take it. I have no idea. What kind of technique do you use to detect such structures?
Yeah, so we've been using mostly manufactured materials, like the kind of magnetic beads you might be familiar with from antibody assays. So these are on the range from 1 to 60 microns. And this is, you bring up a really good point, is we're really trying to understand how much the size of our target substrate will affect our aptamer binding. Part of that is because even if you're talking about a 15 mer aptamer, 15 oligo size DNA strand, that's still going to be probably much bigger than your binding site on your cell or mineral, even at a 60 micron size. That's going to represent a big footprint on there. And then if you add in the need for PLA to give you an easily sequenced binding strand, you're really starting to limit the size of cells or particles that you could interrogate with that method. So that's something we're actively working on, is understanding the size relationship between our particles and the efficacy of this method. This is really what brought us to thinking about theoretical understandings of very small cells that are hard to observe. And this idea of the need for possibly something like cytometry in off-world applications is becoming really important, we think.
Super. Thank you very much.
Yeah, thanks. We should have a paper coming out, I'm hoping later this spring, on this topic, with all of the information about the size, the chemistry of the target, the aptamer that we were able to apply, and how it mapped out in statistical space.
Great. I'll be looking for it.
Okay. Then next question, if somebody wants to ask one.
May I ask a question?
Yes, please. Yes, please do.
Giuseppe Murante from Italy, Trieste. How much of this approach do you think could be applied to distant systems such as exoplanets?
Yeah, so nothing that we're working on can be in our group can be applied necessarily to exoplanets with the tools that we're building, because our first one needs a tandem mass spec, the next one needs sequencing and all of this gadgetry. The third one needs SIMS, that's pretty big. And the fourth requires electrical chemical sensors. So none of these tools that we're building can. Some of these ideas can be applied to the kind of spectroscopic remote sensing data you can get about exoplanets. And I should give a shout out to one of our researchers in lab, Agnostic Biosignatures, Sarah Walker at Arizona State, just got funding for an independent group to start thinking about expanding these applications for exoplanets. This is very fresh work. I think she just started getting funding this month. So if you're interested in how these sorts of ideas of complexity, complementarity, energy distribution, and elemental abundance will apply to exoplanets, be looking at Sarah Walker's work in the future. And because she's part of our group, we have a lot of discussion about this.
Thank you.
Okay, other questions, please?
Hello, can I make a question?
Yes, please.
So first of all, thank you for the great talk. I'm Barbara Soares from Portugal, and I just had a small question. For the many hypotheses on the solar system that you talked about, so Mars, Enceladus, Titan, and so on, which one do you think will be the most promising to get the better results?
Wow, I love this question because it's kind of like asking who your favorite child is. I have to say I have a lot of hope for the ocean worlds, and part of that is just really based on the work I'm doing with people in deep subsurface systems, where I've really started to respect the ability that life has to navigate very low energy systems. And part of that is also because I think of the fact that, our world is an ocean world, and we have, long heritage of life on our planet. It started rising very early in our history, and these are other ancient oceans. So they've been experiencing that same sort of chemistry that you get in liquid environments. Poor old Mars has been stunted by the lack of that kind of chemical availability that you get when you have a solvent embedding all of your chemistry. So I'm not ruling Mars out, but I think Europa and Enceladus are going to be great targets in the future.
Okay, thank you.
Okay. Other questions?
Oh, I hope I didn't offend anyone who's very fond of solid state chemistry by saying Mars is a little bit less great for its lack of liquid.
So if this is not the case, then I would like to conclude this seminar and ask and thank Heather again for coming. You also have noticed that all the talks of the EAI seminars are available on a special YouTube channel, which we sent up. So you just have to look at either at the European Astrobiology Institute website, or to follow the link, which is actually in the chat given by the European Astrobiology Institute, which you just look at the chat and you find the link of the seminars.
Great, thank you so much.